Good morning, class. So today we will cover the formation and secretion of saliva. We will learn about myoepithelial cells, the histology of the ducts and how it affects the composition of the saliva. So the formation and secretion of saliva. The formation of saliva occurs in two stages. The first stage is cells of the secretory end pieces and the intercalated ducts produce the primary saliva, which is an isotonic fluid containing most of the organic components and all of the water that is secreted by the salivary glands. In the second stage of secretion, the primary saliva is modified as it passes through the striated and the excretory ducts, mainly by reabsorption and secretion of the electrolytes. So the final saliva that reaches the oral cavity is hypotonic. The macromolecular components are abundant, abundant rough endoplasmic reticulum and a large Golgi complex store their products in membrane-bound granules in the apical part of the cytoplasm. The secretory proteins are synthesized by ribosomes attached to the cisternae of the endoplasmic reticulum and translocated to the lumen of the endoplasmic reticulum. Proteins are transferred by small vesicles to the Golgi complex, where they undergo further modification and they're followed by condensation and packaging into a secretory granule, which then moves towards the apical part, towards the lumen. The secretory granules are stored in the apical cytoplasm until the cell receives an appropriate secretory stimulus, which is when they are released in the lumen. The granule membranes fuse with the cell membrane at the luminal surface, and the contents are released into the lumen, and the process is called as exocytosis. The salivary glands, the sympathetic neurotransmitter norepinephrine usually is an effective stimulus of exocytosis. Norepinephrine binds to the beta-adrenergic receptors on the cell surface. These receptor activation through G proteins stimulates the adenyl cyclase to produce cyclic AMP. The increased cyclic AMP levels then activates the protein kinase A, which phosphorylates other proteins in a cascade that eventually leads to a granule exocytosis. The fusion of the granule membrane is mediated by formation of protein complexes involving protein cell membrane and proteins of cytoplasm. Release of granule content, the granule membrane is internalized by cell as small vesicles, which may be recycled. Now, another important cell is a myoepithelial cell. These cells are contractile cells associated with the secretory end pieces and intercalated ducts of the salivary gland. They are located between the basal lamina and the secretory or the ductal cells and are joined to the cells by the desmosomes. Myoepithelial cells are present around the secretory end pieces and have a stellate shape. They are filled with filaments of actin and soluble myosin, and that's why they are contractile in nature. Cell membrane has numerous caveolae, which presumably function in initiating the contraction. Cellular organelles are located in the perinuclear cytoplasm. This is the structure of a myoepithelial cell. It's a stellate-shaped cell, centrally placed nucleus. The perinuclear region has all the rest of the organelle. And this is the secretory end piece, and the myoepithelial cell, which is stellate in shape, is around the secretory end piece and near the basal lamina of the base of the cells. You can see here, this is the cell of the secretory end piece, the basal lamina, and under it is the myoepithelial cell. The myoepithelial cell associated with the intercalated ducts have a more fusiform shape with fewer processes and tend to be oriented lengthwise along the duct. The myoepithelial cell provides support for the end pieces during the active secretion of the saliva and also may help to expel the primary saliva from the end piece into the ductal system. When they contract, the saliva from the end pieces go into the intercalated duct. Myoepithelial cell provides signals to the acinar secretory cells that are necessary for maintaining cell polarity and the structural organization of the secretory end piece. Evidence also suggests that the myoepithelial cells produce a number of proteins that have tumor suppressor activity, such as proteinase inhibitor and anti-angiogenesis factor, and these cells may provide barrier against invasive epithelial neoplasms. Coming to the ducts, the ductal system is a varied network of tubules that progressively increase in diameter, beginning at the secretory end piece and extending to the oral cavity. Three classes of ducts are intercalated ducts, striated ducts, and excretory ducts, each with differing structure and function. The ductal system is more than just a simple conduit for the passage of saliva. It actively participates in the production and in the modification of the composition of the saliva. Coming to the intercalated ducts first. These are the ducts which are closer to the secretory end pieces. The primary saliva produced by the secretory end pieces passes first through the intercalated ducts. The first cells of the intercalated duct are directly adjacent to the secretory cells of the end pieces, and the lumen of the end piece is continuous with the lumen of the intercalated duct. This is a picture of an intercalated duct. They are more cuboidal in shape with a centrally placed nucleus. The rough endoplasmic reticulum, mitochondria, and a few secretory granules. The intercalated ducts are lined by simple cuboidal epithelium, and the myoepithelial cell bodies and the processes are located along the basal surface of the duct. The myoepithelial cells are present here, near the base of the cell. The intercalated duct cells have centrally placed nuclei and a small amount of cytoplasm containing some rough endoplasmic reticulum and a small Golgi complex. Few small secretory granules may be found in the apical cytoplasm. The apical cell surface have a few short microvilli projecting into the lumen. The lateral surfaces are joined by apical junctional complexes and scattered desmosomes and gap junctions, and have folded processes that interdigitate with the similar processes of the adjacent cells. The intercalated ducts contribute macromolecular components and include lysozyme and lactoferrin, which is present in the saliva. And other components are also secreted by these cells. Coming to the striated ducts, the intercalated ducts are followed by the striated ducts. They receive the primary saliva from the intercalated ducts and constitute the largest portion of the ductal system. They are the main ductal component located within the lobules of the gland, that is, they are intralobular, and the striated duct cells are columnar in shape, in contrast to the intercalated duct cells, which are cuboidal, with a centrally placed nucleus and a pale acidophilic cytoplasm. The diameter of the duct is greater than that of the secretory end pieces, and the lumen is larger than those of the secretory end pieces and the intercalated ducts. The function of the striated duct cell is modification of the primary saliva by reabsorption and secretion of electrolytes. So the primary saliva is secreted into the intercalated ducts, which then goes into the striated ducts, where it is modified by mostly reabsorption and secretion of electrolytes. The structure of the duct cells reflects its function. You can see here, this is the structure of a striated duct cell. It's columnar in shape. You can see the basal striations of the duct cells here. This is observed when under electron microscope. And these striations are because of the numerous elongated mitochondria. You can see here. These are the elongated mitochondria, and in narrow cytoplasmic partitions separated by highly enfolded and interdigitated basolateral cell membranes. You can see the basal part of the cell has a lot of folds, and between these folds, the entrapped cytoplasm contains these elongated mitochondria, which gives a striated appearance on an electron microscope. And these are the secretory granules which are present near the lumen of the duct. The duct cells also contain numerous lysosomes and peroxisomes and deposit glycogen frequently. Deposits of glycogen are frequently present in the perinuclear cytoplasm. Adjacent cells are joined by well-developed tight junction and junctional complexes. These striated ducts are then followed by the excretory ducts, which have the largest diameter of the lumen. These ducts are located in the connective tissue septa between the lobules of the gland, that is in an extralobular or interlobular location. They have a pseudostratified epithelium with columnar cells extending from the basal lamina to the ductal lumen and some small basal cells. The smaller ducts join to form larger excretory ducts. The number of basal cells increases and scattered mucous cells may be present. In the smaller excretory ducts, the structure of the columnar cell is similar to that of the striated duct cells. As the ducts increase in size, the number of the mitochondria increase. The basal cells have numerous bundles of intermediate filaments and are attached to the basal lamina by prominent hemidesmosomes. In some instances, basal cells may contain abundant of actin filaments. Now, how does the duct modify the composition of the saliva? The striated and the excretory ducts modifies the primary saliva, which is produced by the end pieces and the intercalated ducts. This occurs principally, as mentioned before, through reabsorption and secretion of the electrolytes in the saliva. The luminal and the basolateral membranes have abundant transporters that function to produce a net reabsorption of sodium and chloride ions, resulting in the formation of hypotonic final saliva. The primary saliva is isotonic, but the final saliva is hypotonic. The ducts also secrete a potassium and bicarbonate, but little, if any, secretion or reabsorption of water occurs in the striated and the excretory ducts. The major secretion of water occurs in the primary saliva. The final electrolyte composition of saliva varies depending on the salivary flow rate. So, at high flow rates, when the saliva is in contact with the ductal epithelium for a shorter time, when the flow of the saliva is faster, the sodium and the chloride ion concentrations rise and the potassium concentration decreases. Whereas at low flow rates, when the saliva is in contact with the ductal epithelium for a longer duration of time, the sodium and the chloride ion concentration decreases and the potassium ion concentration increases. So electrolyte reabsorption and secretion by the striated and the excretory ducts is regulated by the autonomic nervous system and also by mineralocorticoids produced by the adrenal cortex. The histology of the major salivary glands now. Now, parotid gland is one of the major salivary glands, and it has spherical secretory end pieces which are serous in nature. So there are serous cells. The pyramidally shaped acinar cells have a spherical, basally situated nucleus and surrounded by a small central lumen. The basal cytoplasm stains with basophilic dyes, and the secretory granules in the apical cytoplasm usually stain with the acidophilic dyes. The fat cell spaces often are seen in the sections of the parotid gland. The intercalated ducts are numerous and long in the parotid gland. They are lined with cuboidal epithelial cells and have lumina that are larger than those of the acini. The striated duct cells are numerous and appear as slightly acidophilic, round or elongated tubules of larger diameter than the end pieces. The ducts consist of a simple columnar epithelium with round centrally placed nuclei. Coming to the submandibular gland, the submandibular gland contains serous end pieces and mucous tubules capped with serous demilunes. Thus, it is a mixed gland. It has both serous and mucous acini. The proportions of serous and mucous secretory end pieces may vary from lobule to lobule and among individual glands as well. The serous end pieces are similar in structure to those found in the parotid gland. That is, they are pyramidal in shape. The serous cells are pyramidal in shape with a centrally located lumen. The apical part has secretory granules and the nucleus is spherical and basal placed. And it has a basophilic cytoplasm. The nucleus is compressed against the basal cell membrane and contains densely stained chromatin. The lumina of the mucous tubules are larger than those of the serous end pieces. Serous demilune cells are similar in structure to the serous NP cells, but discharge the secretion into small intercellular canaliculi that extend between the mucous cells to reach the tubular lumen. The sublingual gland. Now, the sublingual gland is also a mixed gland, but the mucous secretory cells predominate. The mucous tubules and serous demilunes resemble those of the submandibular gland. Although serous end pieces may be present, they are rare, and most structures appearing as serous end pieces probably represent sections through demilunes that do not include the mucous tubule. The intercalated ducts are short and difficult to recognize. The intralobular ducts are fewer in number than in parotid or submandibular gland, and some ducts lack the infolded basolateral membranes characteristic of the striated ducts. Now, the histology of minor salivary glands. The minor salivary glands are found throughout the oral cavity except in the anterior part of the palate, that is the hard palate, and in the gingiva. These are aggregates of salivary end pieces and ducts, which are organized into small lobule-like structures located in the submucosa or between the muscle fibers of the tongue. Usual situation in minor salivary glands, situation of the minor salivary glands are the lingual serous glands in tongue below circumvallate papillae, which are pure serous glands. Their secretions are released in regions, troughs surrounding the circumvallate papillae, and in clefts between the rudimentary foliate papillae on the sides of the tongue. Coming to the clinical considerations of the salivary glands. So the age changes with the salivary glands. First, with age, a generalized loss of salivary gland parenchymal tissue occurs. The lost salivary cells often are replaced by adipose tissue. Although decreased production of saliva often is observed in older people, whether this is related directly to the reduction in parenchymal tissue is not clear. Salivary glands may be influenced by a number of diseases, both local and systemic. Several viruses such as cytomegalovirus, Epstein-Barr virus, and human herpes virus infect and replicate within the salivary gland cells and are shed into the saliva. Viral infections such as mumps and bacterial infections of individual glands may cause inflammation, resulting in a painful swelling of the glands. Ductal obstructions may result from the formation of styloliths or stones in the duct. Most common is a submandibular gland stylolith. It's found in the submandibular duct, or a mucus plug, or the severing of the duct of a a minor salivary gland by trauma causes it. So these are the clinical considerations of the salivary gland. With this, we end our class here. Thank you.